Hello friends, in this video we are going to discuss about differences and similarities between prokaryotic and eukaryotic translation. The translation process is a fundamental cellular process to synthesize proteins from messenger RNA. The primary goal of translation in both prokaryotes and eukaryotes is to maintain the supply of proteins. Still, there are many differences between prokaryotic and eukaryotic translation. So let us see the differences one by one. Prokaryotic translation and transcription process is simultaneous or synchronous. Messenger RNA formed in prokaryotes after transcription can immediately undergo translation. Eukaryotic translation and transcription process is discontinuous. In eukaryotes, messenger RNA formed after transcription is first processed and then it can undergo translation. Prokaryotic translation occur in cytoplasm. In prokaryotes, transcription also occur in cytoplasm, while in eukaryotes, translation occurs in cytoplasm but transcription occurs in nucleus. Prokaryotic translation is cap independent. Eukaryotic translation is cap dependent and cap independent. Prokaryotic translation is performed by 70S ribosome. 30S and 50S subunit together form 70S ribosome. Eukaryotic translation is performed by 80S ribosome. 40S and 60S subunit together form 80S ribosome. In prokaryotes, messenger RNA is unstable. Its lifespan is few seconds to 2 minutes. In eukaryotes, messenger RNA is stable. Its lifespan is few hours to days. In prokaryotes, messenger RNA is polycistronic, can produce more than one peptide. Hence, messenger RNA acts as the template for the synthesis of many polypeptides. In eukaryotes, messenger RNA is monocistronic, hence can produce only one peptide. It acts as the template for a single polypeptide. Prokaryotic translation has no definite phase of occurrence. Eukaryotic translation occurs in G1 and G2 phase of the cell cycle. Prokaryotic translation occurs on the ribosome present in the cytosol. Eukaryotic translation occurs on the ribosome present in the cytosol and rough endoplasmic reticulum. In prokaryotes, first amino acid added to the peptide is formilated methionine. In eukaryotes, first amino acid added to the peptide chain is methionine. Initiation codon in prokaryotes is usually AUG, occasionally GUG or UUG. In eukaryotes, initiation codon is AUG, occasionally GUG or CUG. In prokaryotes, smaller subunit of ribosome binds to Shine-Dalgarno sequence on messenger RNA. In eukaryotes, smaller subunit of ribosome binds to Kozak sequence. So, in prokaryotes, smaller subunit, that is 30S of ribosome, recognize the Shine-Dalgarno sequence in the messenger RNA during translation initiation, while in eukaryotes, smaller subunit of ribosome recognize the 5' cap of messenger RNA during initiation. Prokaryotes may have many start sites and Shine-Dalgarno sequence all along messenger RNA. Eukaryotes always have only one start site, which is located towards the 5' region of the messenger RNA. In prokaryotes, there are three elongation factors, IF1, IF2 and IF3. In eukaryotes, there are 9 elongation factors denoted as small EIF, where small e stands for eukaryotes. They are EIF1, 2, 3, 4A, 4B, 4C, 4D, 5 and 6. In prokaryotes, elongation factors are EFTU, EFTS and EFG. In eukaryotes, elongation factors are small e EF1 and small e EF2. In prokaryotes, release factors are RF1 and RF2. In eukaryotes, release factor is ERF. In both of these, small e stands for eukaryotes. In prokaryotes, process of translation is faster. It occurs at the speed of 20 amino acid per second. In eukaryotes, process of translation is slower, occurs at the speed of 1 amino acid per second. In prokaryotes, post-translational modification of peptide chain occurs in cytoplasm. In eukaryotes, post-translation modifications of peptide chain occurs in endoplasmic reticulum and Golgi apparatus. In prokaryotes, only the formyl group from the first amino acid methionine is removed from the polypeptide after protein synthesis. In eukaryotes, usually the unformylated first methionine as such is removed from polypeptide after protein synthesis. Now let us see similarities between prokaryotic and eukaryotic translation. Both prokaryotes and eukaryotes uses messenger RNA template for translation. In both prokaryotes and eukaryotes, messenger RNA is synthesized from the genetic molecule, which is DNA. Ribosome is the protein synthesis machinery in both groups. All the 20 amino acids are same in both groups. All the 61 codons are similar in both the groups. In both groups, protein synthesis occurs in cytoplasm. In both groups, translation is completed in 3 steps: initiation, elongation and termination. The process of peptide bond formation is similar in both groups. In both groups, transfer RNA picks up and bring the correct amino acid. Peptidal transferase activity is done by ribosomal RNA molecules of larger subunit of ribosome in both prokaryotes and eukaryotes. All 3 stop codons are similar in both groups. In both groups, proteins undergo post-translational modifications. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.